Tim Walsh. I'm a veterinary pathologist here at the National Zoo, and I've got what I believe to be one of the dream jobs for a veterinary pathologist. There's very few large zoos that employ an actual pathologist, and many people don't even understand why a zoo would need a pathologist. They don't want to take the thought that, well, all of these animals are going to die eventually, and we need to get the most information we can from them and continue learning from them so that we can help those that are still in the wild. And so I guess that's why this is a dream job, in that it gives you an opportunity to work with amazing creatures and to translate that kind of knowledge into helping those in the wild or furthering conservation status of wild populations. One of the diseases we work on at the National Zoo is elephant herpes. And elephant herpes was first described here at the National Zoo by Laura Richmond in 1999 using case material from one of our elephants that died of the disease. And this is one of those classic diseases that serve as a large intersection of both different scientific disciplines, including pathology, veterinary medicine, molecular diagnostics, toxicology, and virology. All of those types of disciplines have played a role in it. And in particular for elephant herpes, molecular diagnostics have proven invaluable. This is because nobody has been able to culture this virus to any extent so far in the laboratory. And so all we have to work with is the DNA samples.
I'm Erin Latimer. I'm the lab manager of the Elephant Endotheliotrophic Herpes Virus Lab at the National Zoo. I do the research and diagnostics on EHV. I mostly work with samples from the North American elephant herd, but I also get samples from around the world. The part I like most about my job is that I get to help labs in other countries do the same kind of testing. The zoo is really into capacity building, going to other places and training them to do the testing. We don't like to just go and bring samples back here for testing. We like to train other people so that they can do the testing there on their samples. So I have helped set up a lab in India. It's now called the Wildlife Disease Research Laboratory. And in addition to testing for EHV in the wild in elephants in India, they're also testing for other diseases too that are relevant to Indian animals.
When Laura Richmond and Gary Hayward first really started working, Gary Hayward is our main collaborator and one of the lead researchers for elephant herpes at Johns Hopkins. Once they started working on this using routine PCRs, that was pretty much cutting edge for that day. We came to rely on the conventional PCRs to diagnose this disease. And what we do is accept samples from any elephant herd, either in the United States or internationally if we can get the appropriate permits to move samples, to do diagnostic testing whenever they think they may have a sick elephant. However, in the last 5 years, as more cases have been identified and much of the genetic evaluations have gone on, we've realized that this elephant herpes, which we always kind of talk about as one virus, is really at least 8 different viruses with many different subtypes. And so that really makes evaluation of the genetic material much more important, provides much more information in regards to how the elephants are interacting with the virus and the epidemiology of the virus within the herds.
There are now 7 known EHVs, and some of them are quite different from the others. So the next-gen sequencing will help us compare the different EHVs and figure out why their genes are different, why their pathogenesis is different. Some of the genes are so different that we're not able to pull them out of some of the viruses because we can't design primers to them. The genes are just too different. With conserved genes, it's easy to design primers and pick up genes from other related viruses. But some of these viruses are so different from the other ones that primers can't be designed. So the long reads will help. Right now, the one that I'm working on, it's a piece of the EHV2, and it's going to help us figure out some of the latency genes they're involved in the herpes viruses going latent in the elephants.
It's not only a disease of captive animals, but is also prevalent in wild animals. And one of the research pushes now is to gather more samples from both captive animals and wild animals to really delineate this relationship between these different types of viruses and how they interact. And because elephant populations in the wild, just like in captivity, are becoming more and more fragmented by loss of habitat and ecosystem pressures, these types of research questions will have great implications for further management of elephants as a resource and as a valuable member of biodiversity.